Hello friends, this is Dr. Jyoti Saxena and I welcome you to my channel Biotech Medicine. The biotech products like the vaccines, insulin, hormones and different type of enzymes, they do not go directly from lab to the market. It's a long journey. So let's find out step by step. There are many phases during this journey, and the first phase is the research phase. This is the phase of idea and discovery stage, you can call it, because this is the time when the ideas come and then you start doing the discovery. Everything you can say starts with a problem. If there is a problem, then people start looking for the solution. So suppose we take the example of diabetes. If there is diabetes which is prevalent in the society, then people started thinking about the solution, and the solution came in the form of insulin. Similarly, if there are infections, then you need antibiotics, and if you need the eco-friendly agriculture, because the chemical fertilizers and chemical pesticides, these are causing lots of hazards to the environment and for the human beings. So that's why you need biofertilizers. So that's how the problem was there first, and when people realize that problem, then they start looking for the solution. So that's what the scientists do. They quest for the solution. So scientists, first they identify the target molecule or the gene depending upon the product. And then not only this, but they have to study the mechanism. You should know everything, each and every aspect about that target molecule or the gene. And then the scientists perform the lab experiment. Of course, initially these are done at the small scale only. And this stage can take years, because it may look that it's a very simple stage. The first phase should be very simple one, but it may take long time, and many scientists in different countries, in different labs, they publish their results, and based on those results, the researchers proceed. So next phase is the proof of concept. That means this is also a lab scale development. Here we go from concept to the prototype. And prototype, you can say, the first version of the final product. And here the prototype should be reproducible. You should give the results which are reproducible. That means that those can be repeated. Then only it is successful. Otherwise it is a waste. And for this, the infrastructure and technologies are required, of course. Different type of equipments are needed. Lots of money is also needed to do this kind of work. And from small quantities to the process optimization we go. And process optimization is upstream. Here, we optimize the media composition, how the cell culturing is done. So that's all the conditions are optimized. So upstream as well as the downstream processing is done. Downstream is how the purification is done, how you harvest the product. So that all is done before we go to the large scale production. So at the lab scale, but at the small scale, we have to do this first. And here we have to see the purity and potency and safety of the product, because ultimately this product is going to be useful for the human beings. Human beings are going to consume it. So that's why the potency and the safety of the product, that is very important. And designing the protocols for having scale up possibilities, whether it can be scaled up or not. So that again is very important here, because many experiments are done at the small scale, but they cannot be scaled up. So with these kind of biotechnological products which come to the market, this is not possible. So scale up possibility should be there. And for that, we go from flask level to the bioreactor level. Of course, the initial level bioreactors are very small bioreactors, the lab scale bioreactors. But we have to take our experiments from petri dishes and flasks to the bioreactors. And at this level, people or the scientists get different patents for processes, for the products. So these patients are very helpful, and these are very important also. Then starts the preclinical trial. This is mandatory phase. You can't do the clinical trials on the human beings directly. So for that, preclinical trials are mandatory. So here the in vitro and in vivo studies are done. In vitro means the experiments in the petri dishes, in the flask, that kind of experiments are done. And then in vivo studies which are done on the animal models. So these are tested on the cell cultures, animal models. Here we have to assess the safety and efficacy, where this product which you are making, that is efficient or not, whether it is safe for the animal models or not. So that has to be reported here. And many products do not pass this phase, and that is, at the experimental level in the lab, it's okay. You are getting good results, you are publishing the results. But then if it is not safe for the animals, then it cannot be safe for the human beings. So this is something which is important. It may take 1 to 2 years. Actually, in all these steps, in all these trials, lots of time is consumed. Then comes clinical trial. Now, these clinical trial is something which are done on the human beings. Human testing is done, and there are 3 phases. If we see the phase number 1, here only the small group of 20 to 80 volunteers are taken. These volunteers are healthy volunteers, and before they are used for this phase 1, they have to sign a consent letter because there is lots of risk involved. These are the human beings, volunteers, which are for the first time the product is tried on these human beings. So that's why lots of risk may be there. So here, because these are healthy individuals, and all the tests, ECG and blood related tests, every kind of test is done before they are chosen. So here the safety parameters are and the doses, and whether there is any side effect which is seen on these healthy individuals. So that is all determined here. So this is phase 1. Now comes the phase 2. This is a little larger group. 100 to 300 patients are here. But here, see, these are the patients. These are not the healthy individuals. And these patients are recommended by the doctors, or they are there in the hospitals. Many times, through social media also these patients are selected. And here the studies are done related to the effectiveness and the further safety assessment. So whether the product is effective on the patients or not, and whether it is safe for the patients or not. So those kind of studies are done here. And now the phase number 3, that is done on the large scale. Patients, thousands of patients are taken here. All the studies are done in the controlled way. And this is a kind of confirmatory phase, because here the efficacy, whether it has any adverse effect or not, those are monitored against the standard treatment. For any disease, for which we are going to use this product, there might be some standard treatment procedure also. So we have to compare the two, whether this product is better or not, what are the safety measures, what are the, what is the efficacy and all that. It has to be seen here. These clinical trials are very, very expensive. They are highly regulated also, and you see the time period. 5 to 7 years may take for completion of this phase. So lots of time is consumed, and millions of dollars go into this kind of a study. So this, you can say, very long period is required for this clinical trial, and also lots of money is required. Now the regulatory approval. This is very crucial phase, because without approval you cannot go into the market. So whatever data we have got until now through the preclinical trials and the clinical trials, that data is compiled, and that is then submitted to the different regulatory authorities for the licensing and also for the new drug application. FDA is in USA, European Medicine Agency is in Europe, and Central Drug Standard Control Organization is the authority in India. So these are the major regulatory authorities. These authorities review the safety data, whatever data you have submitted, efficacy data, good manufacturing compliances are also seen, how the manufacturing is going, what is the raw material, which is whether it is available or not. So all that is controlled here. It is monitored here. So after that only the approval is given. Then comes the manufacturing and quality control. Once you have got the approval, and then the commercial scale production starts. Before the approval, you cannot go for the commercial scale production. So, and most of these products, they are, released batch wise. So batch wise testing and sterility testing, whether they are stable or not, and packaging validation. So everything is done batch wise. It's not that in one batch the regulation is enforced, and for the next batches it is lenient. No, it's not like that. For each and every batch you have to be careful, and you have to follow all the regulations, and each batch must meet strict standards. And then comes the market launch, and after that the post market surveillance is also there. So once it is launched into the market, the product is launched. Even after that also, the monitoring is done to see whether there is any kind of allergic effects, or whether there is any kind of adverse effects of this medicine. And in that case, the modifications are done. So you may call it a phase 4. Now let's see what are the key factors for the success. What are the key factors? That a product which was conceptualized in the lab has come to the market. So what are the chances of success? And it depends on what. First thing is the financing, the seed and venture capital. At both the times, when the concept started for the lab scale experiments, you need money. And also for the venture, at the large scale, when you are going towards the clinical trial and the marketing, for that also, you need lots of financing. And believe me, millions of dollars are needed for a product to reach to the market. Then scalability is another important thing. When we are doing the research in the lab conditions, the product may be very effective, and we may feel that it is going to be a very revolutionizing kind of product. But if it cannot be scaled up, it cannot be produced at the large scale, then you cannot have the success. So scalability of that product is very important. Then regulatory approval. Without approval of the product, you cannot go into the market. So this is a key factor for the success, whether the regulatory authority is approving it or not, and whether it is approving it at the right time or not. If the time taken by the regulatory authority is extended for a few years, then lots of money is wasted. And then it should be of at the affordable cost, because ultimately the patients are going to buy that. So it should be affordable. Not everyone can buy very expensive products. So at last we can say, this journey is a very long journey. From petri dish to patients, that means we start the concept at the petri dish level, at the research level in the lab. Then there are many steps. One by one, it goes to the patient and then to the market. So it's a long journey, and this is the journey of biotechnology. Each and every step, biotechnology is involved. And you can see these days the biotechnology, recombinant DNA technology, genetic engineering, all these fermentation processes, they are revolutionizing our medicine. So thank you.